So, there is another method. So, here they use the energy based criteria. So, mainly they calculate the van der Waals energy and the electrostatic energy, right. Then using these interactions, right, you can identify the binding sites. How to identify the binding sites? For example, if you have these PDB coordinates, right, in all the cases we use the structures, right. In this case you can take these binding sites as a kind of experimental data, right, because we are not doing any experiments directly to identify these binding sites or binding affinity, but we use the known 3D structures, right, and use this criteria to identify the binding sites. Fine. So, how to identify the binding sites using energy based criteria? So, here this is the chain A and chain B. For example, if you take the arginine 88, right. So, there are different atoms in arginine. So, for each atom you calculate the energy with all atoms in the partner chain, right, for all atoms in the partner chain. So, then you do with the second atom, in the third atom, so they calculate the energy, then sum up all the energies in all the atoms in the arginine, then you sum up the values, that will give you the energy of this arginine 88, right. If you want to take the arginine 88, take all the atoms, with each atom in chain A, we calculate the energy with all atoms in chain B, right. Do it for all the atoms, for example, N, Ca, Co and so on. Then we sum up the values, that will give you the energy of arginine 88 in chain A. Likewise, you can do for all the residues, right. For example, 100 residues, so you get 100 energy values. From these energy values, you can identify the binding sites based on the energy, right. So, how to do that? This is an example. For example, the 1AI4A tryptophan 74 has minus 13.2 kilocalorie per mole, right. Like this phenylalanine 211, it has 14.78 kilocalorie per mole, right. Fine. So, now from these energy values you can see which residues are high preference to interact with the binding with the partner. So, we have the energy values, we divide in different bins, right. For example, minus 2 to minus 1.9, minus 1.9 to 1.8, right. So, each bin you can see the frequency of occurrence of the residues with a particular energy, right. For example, if you see minus 2 to minus 1.9, right, minus 1.9 to minus 1.8, so we can go with up to plus 1, right. So, here there are two different colors, one is in blue and one is in green. The blue one gives the data for each bin and the green one gives the cumulative values, right, because we can go even less than minus 2, here I show the value, many values less than minus 2, right. So, this is where if it is less than minus 2, you can see that is around 7.7%, because the bins are smaller, this is why I did not plot that. So, finally, if you see minus 2, there are very less number, right, then up to minus 0.5 you can see the rise, right, and there is a peak, right, and then it is going down. So, if you take a cutoff of 1 kilocalorie per mole, for example, take this cutoff, right, then how many residues, so what is the percentage of residues which are identified as binding sites? If you do this, there is about 10.8% of residues, right, which are identified as binding site residues, right. This is reasonable, there is relevant to the reported literature. And another case, if you see there is a peak, right, for example, take the energy of minus 0.3 to 0, there is 77%. Why this is a peak here? Because the energy values is very less, right, it is not significant. So, in this case, if the two atoms or the two residues which are far away in the structures, they are not making any contacts, right, they are not making interactions, that is the reason why the values are around 0, right. That is about 77% of the residues, they are not in contact with each other, right, fine. So, here we get the data based on the energy, right. When you compare the data with the distance, because energy and distance are almost similar to each other, right, because energy values are computed with two types of interactions. What are the interactions we used? Coulomb's law as well as this van der Waals, right, both are based on distance, right. So, some advantages, some disadvantages, right. Now, if you see the energy, you can see about 10.8% of residues which are identified as binding sites, and the distance we use different types of distances, you can say if you use the 6 angstrom with any atoms, right, you can say about 13.6. If it is C alpha, it is 4 and it C beta it is 7, right. So, our 1 kilocalorie per mole it is in between the C beta distance as well as with any atoms. And if you see there are many mismatches, about more than 80%, 90% you can see the matches, and some mismatches mainly because of the some of the repulsive interactions are also taken into account in this type of energy based criteria. That is the reason. Right, now I give you an example, right, how to get these interactions, right. So, here these are the values, right, the energy values with some residues, right, energy values, and if you look into the structures, right. So, for example, I show one example in 127, this arginine 1112 and this is tryptophan 144, right. These two residues they try to interact. So, they are interacting with an energy of minus 6.2 kilocalorie per mole, right. This is the positive charge and this is the aromatic ring, right. So, they are making a set of interactions. Which interaction they make? Cut and pi interactions, right. This is the pi system, this is the positive charge. So, they make cut and pi interactions, right. Then the question is, there are some specific interactions, are there any preference of amino acid residues to form this type of interactions, right. So, in this case I show the propensity of different residues to be involved at the binding interface. So, in this case we just calculated the binding propensity of this residue I, right, using the number of residues of type I in the binding site, right, with the normalized with the number of residues in the whole protein. So, what is n bind of I? Right, for example, if we show this figure, right, I show this figure, right, how many residues of type I at the interface, this is the interface residues, for example, alanine, how many alanine in the interface compared with the all alanines in the particular protein, right. So, if you do this, right, you can see some preferences, for example, the arginine which has a value of 15.1 and the tryptophan 17.28 and the tyrosine 15.33, you can see phenylalanine also very high, right. So, you can see the preference of some specific residues, right. From these residues you can see that there are some sort of interactions, right, they are preferred to be at the interface due to the type of those residues, right. Now, the question is, here we have only one residue propensity, right. How can we estimate, how can we identify the interacting partner to identify the type of interactions? In this case what do you need? We need the partner, right, because the binding propensity will tell you the which residues are preferred to be interface, like arginine or tryptophan and tyrosine. To understand the type of interactions, right, we need the partner. So, for example, tyrosine or arginine, right, trying to interact with which type of the residues, right. In this case you take these residues in this complex, right. For example, this residue and this residue interact, this is which type of residue and this which type of residue, right. For example, this could be the phenylalanine, right. Here you can see tyrosine, then you can see both are in the ring system, they can by pi interactions, or here you can see the arginine here, right, and here you can see the tyrosine here, then you can see which type of interactions they can make, right. So, in this case we need the pairs, right. How to identify the pairs, right. So, you can see the preference of I comma j, right, to using this equation n I j, this is number of types of I paired with j, I from the protein 1 and j from the protein 2 in the partner, and Ni is the number of types residue I and j is the number of residues of type j, right. When you look into the binding partners you can see some preferences. For example, you see aspartic acid and arginine, right, you can have the highest value of 8.64, then you can see the arginine tyrosine 6.1, right. Likewise, tryptophan tyrosine 6.5, tyrosine tryptophan 8.1. So, interestingly, if you see these numbers you can find some sort of interactions are important. For example, if you take aspartic acid and arginine, Which type of interactions are important? Electrostatic interactions, right? This minus, this plus. Also it does not depend on the type of the protein, whether you take 1 or 2. Even if you take the other way around, that is also very high, arginine and aspartic acid. If you see arginine and tyrosine, this is a ring and here this is the positive charge, right. And if you see the other way around, you can see tyrosine arginine also having high preference. Then tryptophan tyrosine, this is also pi and this is also pi, you can see the pi interactions, right, aromatic interactions, right. Now the question is, okay, you identify the residues which are at the interface and you identify the pairs, right, from the partner proteins, protein 1 and the protein 2, right. So now the question is whether these interactions are specific or non-specific. How can you estimate whether these residues from electrostatic interactions or not? Right, then we need to see whether the energetic contributions are from the main chain or from the side chain, right. If the contributions are mainly from the main chain, then we can, this is not specific. If they are from the side chain, then they are specific, right, fine. So in this case we can see the contribution from the main chain atoms and the side chain atoms, right, and we compare the values in the main chain and side chain, you can see side chain values are at least twice, right, than that of these main chain atoms, both in the case of the protein 1 or the protein 2, right. Here I define ligand as the smaller portion of this complex and the receptor as the bigger part of this complex, right. So whatever this you consider, you can see the side chain contribution is higher than that of these main chain atoms. In this case you can see these interactions are very specific, right. Now we need to experimentally verify whether these residues they form typical interactions or not, right, fine. So in this case we need to identify the residues which are serving as the hotspot residues, right. So in this case we put a cutoff of 2 kilocalorie per mole, right, we mutate any specific residue. So what is mutation? Change of residues, right. For example, if you change any specific residue, right, for example, you change glutamic acid to alanine, if it changes the binding affinity of more than 2 kilocalorie per mole, then you can see these residues as hotspot residues, right. So then we scan the literature, many data available in the literature, and from this data we can find about 217 types of interactions, right. That means they can reduce the binding affinity, right, at least 2 kilocalorie per mole. So there are many mutants are common. So we take the unique interactions, right, we about 68 interactions, and we look into the residues, which residues are mutated for this drastic change. Most of them are charged residues, about 38 residues are charged residues, and if we take the positive charge in aromatic, there are 32 mutations and the hydrophobic is only 7. This will again confirm that what are the interactions we observed from these pairs, right, aspartic acid arginine electrostatic, cation pi and aromatic interactions, right. These residues are important, right, to form any typical type of interactions, right. So to get more information regarding this type of interactions, right, recently we developed a database, right, this is called the proximity database. This will give you the binding affinity of different mutants, currently it has more than 6000 mutants, right, and we will use this information available in the database, right, to understand, right, which type of interactions are important at different locations, right. Here I generalize with the type of interactions and the specific pairs, right. I will explain a little bit about the database, right, and this contains the binding affinity of protein complexes, right, along with other different types of structural information as well as the literature information, right. We have the search option here. So we use different information, you can search based on the proteins, if you know the complexes, right, protein 1 and protein 2, right, you can get all the data for this particular complex, or if you want to get the hotspot residues, right, you can see the mutant KD or mutant delta G, right. We have the delta G, right, give the range, and you can see the value, the mutants which are serving as hotspots. So, here you can give the mutation type, either you can use from any residue to any residues, right, you can take the information, and you can search with experimental techniques and specific secondary structure, right, helix or strand, and also you can see whether you need only the interface residues, right. Mainly if you see the binding affinity is affected by the interface residues, and hence most of the data you can see they replaced only at the interface region, right, fine. So, you can get the hotspot residues giving the value of 2 kcal per mole, right, delta G values, and get the hotspot residues, and you can also analyze whether this is secondary structure or how it affects in the different functional class or how it affects with the location of residues. For example, you can see the accessible surface area and so on, right. Now, this is the result. For example, if I put the data bovine cationic trypsin and this protein 2, right. So, you can see the inhibitors. So, you can see this is a trypsin, this is a trypsin inhibitor, you can get all the data. So, this is secondary structure is loop, accessible surface area is completely buried, right, and you can see the wild type Kd, right, and the mutant Kd, and have the delta G values, right, you can see all the values. Then we give the PubMed reference, so that you can link to the PubMed, right. What is the PubMed? Literature database, right, and you can also given the exactly where you can get the data, right, table 1 page 177 you will get the exactly the same data from the literature. So, this will help the users to verify the data as well as to get more information regarding any particular complex, fine. So, now we give the more details about the interactions, right. Here there are 2 chains, chain E and chain I. These 2 residues are making the complex, E is making the complex. This is based on the enzyme inhibitor complex, and here this is the data, right. It is a wild type Kd, and we have the mutant values, right, and the delta G is minus 17.95 kcal per mole, right. This is the loop region, and the relative accessible surface area is 0.35, right, that is 35%, the area is 50 angstrom square, right, that is fine. So, when you go to the details, you can get the summary and the complex structure and the interactions as well as the reference. The interactions, if this is your protein, right. So, now you can have what other proteins they are interacting with a particular protein. So, you can have the value from the string database, right. So, and have the all the annotations and what are the residues which are interacting with the particular proteins and so on. So, give the references, these are the authors and the title as well as the details of the particular paper, fine. So, now we give the statistics. Currently we have 176 complexes, right, and the literature resources about 184, and there are more than 5,000 single mutation entries. So, these entries are sufficient, right, to do any analysis. For example, if you want to analyze hotspot residues, or you want to analyze the residues which will not make any changes in the binding affinity, or you can also relate the binding affinity with other features, right, to understand the features which affect the binding affinity, or you can derive models, different methods you can derive to predict the binding affinity upon mutation. We have sufficient number of data, right, in this case it is possible. Then also you can see a double mutation 748. The double mutations you can use and see whether the binding affinity of a double mutation, right, can be explained with the sum of the single mutations, or they are totally different. If it is totally different, what factors influence this difference, and if it is additive and why they are additive. So, you can do the analysis, right, using this data, fine. So, with respect to the functional class, so there is mainly the antigen antibody complexes more than 2000, right, entries, and enzyme inhibitor also about 2000, right, followed by the other classes GC coupling receptor, right, receptor complexes, other enzymes and so on. So, then with respect to the different parameters like delta G, we can have the minimum value of minus 12.2 to the maximum value of plus 12.2, right, and also you can have the different data based on the experimental techniques. So, this is a mutation matrix, right. So, what can you infer from this mutation matrix? Like protein stability, right, protein stability mainly we obtain the data for alanine mutations, right. Here also you can see the alanine mutation, right, because alanine is scanning mutagenesis. So, you can see the plenty of data whether these residues are mutated to alanine. With why there are many data for alanine? Right, because we can see the difference, right, because if you take 20 different amino acid residues, right, glycine is the simplest one, but the problem with using glycine is there is no side chain, hydrogen is side chain, so it is completely flexible. In this case flexibility is also important, right, plays a role, right, for this binding affinity. We take the alanine, so we write because all the 18 amino acids they contain the CH2, right, and the others are different. So, alanine contains CH2 like all the other 18 amino acids, right. So, if you mutate any residue with alanine, you can see the influence or the contribution of different amino acid residues, right. That is the reason why they use generally it's alanine, right, to get the difference in binding affinity, right. So, what are the other mutations which are frequently done? So, they used glycine to serine, right. Okay, then asparagine to serine, this makes sense, right, because asparagine is the polar residue, right. So, we made asparagine to serine to see the binding affinity, and the threonine, right. So, here arginine to methionine, E to E to D, okay. So, if you do E to D, what will happen? Length of the CH2 group, right. So, you can see the how the length around CH2 group reducing 1 from E to D. So, change the binding affinity. Likewise, they did the lysine to arginine, not okay, they increase lysine to arginine, arginine to lysine, not right, right. Which one? Leucine to yeah. Leucine to methane, right. So, there are many mutations. This will tell you how far the binding affinity, right, changes with respect to amino acid mutations, right, fine. So, you can do the average assignment method like here also, assign the values and then see whether we can identify the mutants, right, which increase the binding affinity or less decrease the binding affinity, or whether we can be able to predict the binding affinity. And currently there are several methods are available in the literature, right, but now with the available amount of data, right, we can make a good model for predicting the binding affinity upon mutations, right. So, we give the links, so we link to the different other databases, for example, the protein data bank, this is for the protein structures. What is UniProt? Protein sequence database and the PubMed, this literature database and the string, protein interaction networks. You can see if you have any particular protein is linked with or this is interacting with what are the other proteins, right, in the network. For example, if you construct a human protein interaction network, right, there are many proteins that are making as a hubs interacting with other proteins, that information you can get from the string database. Also, you can get the dictionary of protein secondary structures DSSP, right, from this site, and you can get this binding affinity data from different other resources. One is the AACDB, right, alanine scanning energetic database, right, and the PINT, these are developed earlier, right, and you can see the SCMP database is also contains the energetics of mutation protein interactions. But compared with the other databases, our database proximity has several advantages. We can have the interface and you can get different types of data with the functional classes and more number of data. So, currently it is including the wild type as well as the mutant data, fine, right. So, from this database you can identify the mutants which are termed as hotspot residues. What are hotspot residues? Yeah, at least the binding affinity change by 2 kilocalorie per mole. So, earlier we discussed and we identified some type of interactions, right. Then we will see whether these interactions are important or not, right. I will show a few examples. So, this is a complex C6AP UBC H7 complex. This will tell you the importance of aromatic interactions, right. So, here Elter and Coleman, so they experimentally, right, measured the binding affinity of 49 mutants in this particular protein, right, and they showed that 15 residues are hotspots Among the 15 residues, if you see 10 residues which are involved in cation pi interactions and 5 are involved in electrostatic interactions, right, only 3 are hydrophobic residues. Then specifically if you tell, look at this, replace phenylalanine at position number 63 to alanine, right, they changed free energy change of 3 kilocalorie per mole. Now the question is whether this F63 is important or not, right. Using a structural analysis, we also showed the phenylalanine, right, is important for the binding affinity. So if it replaces this one, that should reduce the binding free energy. So take this complex, structure is known, and get X F53 and how this F53 is interacting with other residues, right. So you can see this phenylalanine 63, right, is interacting with tyrosine 694. Now if we see the contributions, all the main chain contribution and the side chain contributions, and we see most of the contributions are due to the side chains, and you can see the interaction energy of minus 1.2 kilocalorie per mole. In this case, if you mutate this residue, this will change the binding this free energy, and this eventually reflected in the case of these solution studies like the free energy change of 3 kilocalorie per mole. That means if you mutate the phenylalanine, right, change the free energy by 3 kilocalorie per mole, because this phenylalanine is very important, even the structure they are making good interactions, right, specifically aromatic interactions with tyrosine 694. I show another example. This was the cation pi interactions, right. Here the Zhang et al. They analyzed 29 different mutants. Among 29 mutants, 11 mutants are identified as hotspots and 6 are cation pi interaction forming residues, 3 are electrostatic interaction forming residues, that means charge residues, right. They mutated Y127A. This case here change the free energy of 2.2 kilocalorie per mole, that means they are also identified as a hotspot residue. So if you see whether this tyrosine is really important for the binding affinity, then they should make good type of interactions with the binding partner. So if you see here, this is the tyrosine 127, right, this is tyrosine 127. If you look into the binding partners, right, for example in the interleukin receptor 4 and receptor, so this arginine 85, right. So they having the energy of 2.4 kilocalorie per mole. If you calculate the interaction energy, right, so this is 2.4 kilocalorie per mole, this is the van der Waals plus the electrostatic interactions, right. So in this case, these two residues, they are very important to form the interaction, specifically cation pi interaction in this particular protein interleukin 4 receptor, this complex, right, fine. So another example, this is the RAS protein. So here this is the importance of electrostatic interaction. So here this group they mutated 27 residues. Among the 27 mutants, they identified only 6 are hotspots, and if you do the analysis on the hotspot residues, 5 are involved in cation pi interactions and 4 are the electrostatic interaction forming residues, they are charged residues, right. So here they mutated lysine 13 to alanine and they reduced the free energy of 2.5 kilocalorie per mole, and D238A, you can see here also they showed the free energy change of 3.9 kilocalorie per mole, right. If you look at the structure, now this is the ITG studies and we go to the structure. So lysine 32, right, and the aspartic acid 238. So these two residues, so these are the two residues they try to mutate and they are interacting with each other mainly because of this side chain, right, with the free energy of minus 2.5 kilocalorie per mole, they are making very strong interactions. This shows that these residues K32 and D238, they are important to form the electrostatic interaction. This is the reason why they are having the favorable interaction energy of 2.5 kilocalorie per mole, and if you mutate either K or D, they change the free energy of more than 2.5 kilocalorie per mole. That means you can see there is a agreement between the computational approaches obtained from the structures as well as the free energy change, right, they measured from these experimental techniques. So what are the various aspects we discussed today? What different types of complexes? What are different types of complexes? Protein complexes, protein DNA complexes, protein RNA complexes and protein ligand complexes. What are the specific functions of protein complexes? cell signaling, ubiquitination, molecular switching, antigen antibody interactions and so on, right, immunology response and all, right. For the case of the protein nucleic acid interactions, transcription, right, DNA repair, right, packaging and so on. Protein ligand interactions, mainly the acting as an triggering as an inhibitors, right, for any activity, right, and you can also use the protein ligand interactions, right, to identify the inhibitors, right, basically in the case of the structure based drug design or ligand based drug design, right. So, now if you have the complex, it is very essential to identify the binding sites, right. So, how to define the binding sites? So, the two proteins, right, are the two binding partners, whether they share a common interface, right, that region you can see the binding sites, because they are close to each other, they share the common interface area. How to identify the binding sites? Distance based criteria, accessible surface based criteria, energy based criteria. How to identify the binding sites using distance based criteria? Okay, you need the atom coordinates, right, we need exposure coordinates. See the protein 1 and the partner protein 2 and get the distance, right, of any atoms in protein 1 and protein 2, that is very important, right. If we set up any distance, say 5 angstrom between heavy atoms, right, if they are within the distance, you can identify this as the binding site residues, right. So, how to identify the binding sites using accessible surface area based criteria? Reduction accessibility, right, either they have the complex, and if you take out the protein and see whether any change in accessible surface area, right, upon complex formation. If there is a change, then you can say that these residues are involved in binding, right. Then what is how to identify the binding site using energy based criteria? Calculate the energy, like distance you can calculate the energy for each atom and sum up the atoms for each residue and see, right, which residues they have the free energy of -2 kilocalorie per mole, then identify as the binding site residues, right. So, when you identify the binding site residues, either use the distance based criteria or acceptable surface area based criteria or the energy based criteria, you can find the binding propensity, right. So, if you do the binding propensity, which residues are preferred at the to be at the interface? Arginine, and you can see the tyrosine, tryptophan and so on, right. So, then we see whether these residues making any specific partners, right, we identify some partners, right. So, these partners they tend to form some specific types of interactions, for example, aromatic interactions, right, cut and pi interactions, electrostatic interactions. So, we discussed with examples, right. Experimentally they proved that this binding affinity, right, drastically changes if the residues are involved in any specific types of interactions, and if we mutate, right, this will change the binding affinity, right. So, then we discussed about database which contains the binding affinity of complexes and the mutants, right. Which database we discussed? Proximate, right. Proximate contains which type of data? Mainly the binding affinity. So, you will get the binding affinity of any mutation, right. You can try to use these values, right, for any understanding the binding affinity of any complexes or the affinity upon mutations, right. So, in the following classes, okay, we will then move on to the protein ligand interactions and how to use our computation techniques for designing the inhibitors, right, using structure based drug design or using the QSAR techniques and so on, right. And after that we will discuss about some of the common problems, right, and how to approach the problems using bioinformatics approaches. Thanks for your kind attention.